Thank you all for joining, and today we are going to have a session on the current pandemic situation. So everyone is discussing, and all the research community in every research field is thinking about how to contain and how to fight against the coronavirus. So today we have an expert who has worked on systems biology, and of course he's also an emerging researcher on the COVID-19. Some of the pilot studies he has done. Dr. Prashant, he's a senior scientist on a specialization of systems biology in the Department of Bioinformatics from Birla Institute of Science and Research, Jaipur. So today he will be giving a session and his outcome on the pilot studies. But in the meantime, as you all already know that our usual style of presentation, so if you can please be attentive here, that those who have joined via Meet, I would recommend to switch off your webcam so for a better bandwidth, and then you will be in mute mode. But if you wanted to engage in the discussion, please either chat or unmute yourself and kindly tell your name before engaging into the discussions. Most of the questions will be addressed through menti.com. So as I told you, you have to go to menti.com and then just give the number 741474. So you will be able to ask questions in each of the slides, and all the questions will be anonymous. So we entertained you to use menti.com. And the next part will be also the agenda of the meeting will be only decided now. As you can see, I can see a few of them have already started casting their vote. So on the menti.com you will be already having the questions now. You can choose maximum up to 4 area. So this is like if you are interested to know about virus spread, variants, genomics, sequence analysis, immunogenesis, proteomics, or diagnosis. So I can see already 12 people have already voted. So if you want to submit more, you can still do it. So I'll do a countdown up to 5, and you have time until then for me to change the slide. So if you go to menti.com, you will see this page and you will be able to cast your vote. So this just to understand what is your expectation and what you're looking at, and so just to focus on more on those questions and aspects. And then next is what we are going to do is, okay, thank you. I can see that there's a balance between understanding about virus spread, the sequence analysis, diagnosis, and genomics. So this is what I can see. Oh, I can see now more people have come in. Okay, 22% on sequence analysis. Genomics and virus spread is off balanced. Okay, that's great. Now just I'm going to the next slide to understand what is your confidence level on the following areas. I can see few of you have already casted your rating. So you can just slide or click on the rating from 1 to 5. So 1 says I'm not an expert or no voice, and if you are on the rating of 5, you say you are an expert, so that we will be able to see how things goes fine. Yeah, I can see people are submitting their cards. We'll wait for a few seconds so that everybody get a chance to do it. Okay, so I can see here everyone wants to more focus on sequence analysis and understanding of SARS category. Of course, there is SARS virus, MERS, and coronavirus. They are all on a very similar probably category, and Prash will be able to give much more insights on that. So today we will be discussing about the topics ranked top on a different on similar concepts. Of course, Prash will be concentrating more on systems overview, and I have to put a disclaimer. All of you should refer to open source data, and no promotions are being entertained. and all the opinions and discussions are personal view with no connection to our current employees, your current employees, corporates, or any other individuals. So before going to that, so I pass on to Prash. So Prash, you could share your screen and please take the session forward. Thank you so much for accepting this today.
Yep. Thank you so much, Giri, for the wonderful introduction and wonderful or human service that you're doing towards this. And I really like the disclaimer. I think that is certainly the need of the hour.
Yeah.
So I'm just trying to present now. I hope everyone is audible.
Yeah. Yeah.
Yeah, 1 minute, please. I don't know why it shies away. Yeah. Um, so so now I hope everyone can see the screen, Giri.
Uh, it's coming up. I'll tell you once it is up. I'm yeah, it's up now. Yeah,
Yeah.
we can see.
Yeah. So I'll be in the full screen mode. So please just stop me, if something interrupts.
Sure, sure,
Okay.
sure.
Yeah. So friends, thank you so much, and I think this is certainly the need of the hour, and I think given the less spectrum of viral biologists all over the world, many microbiologists or many erstwhile biologists tend to pop up into COVID science. I think this is certainly the need of the hour. So as a scientist or as a practitioner who respects open access, I also thought that I should also do a bit for COVID-19. And together with our team, together with my group, and together with my collaborators and my mentors and my mentees, we just started doing this pilot analysis. And we always try to keep paraphernalia away. When some such pandemics occur, when many people keep on doing a lot of science like this, the most important stuff one needs to look into is keep the paraphernalia away. Means do good science, collaborate more, and not publish very often, but do concrete science so that you can make a good mirror of this. Okay, that is certainly the need of that. If that is not happening, then any country would be in doldrums, because there might be a lot of interactions, a lot of mishappenings, doing this, and above all, it would be a gross injustice, towards the science that we are doing. So we always need to reach consensus in bioinformatics. So if you are analyzing some analysis, always get it analyzed by your colleagues, be it your senior colleague or a junior colleague or whoever it is. Okay, and the most important stuff here is not only stop or not only reach the consensus, but also get it analyzed, get it curate. That is really a very important step. So as a systems biologist, I often ask, what systems aspects would really make a mirror of COVID-19. So these are some of the lessons that we are trying to ask. So one important aspect that we often asked is, what systems aspects are important for a viral study. So the most important stuff with the viruses is they're pretty small, they're omnipresent, and the contagion is all brighter side. Okay, which means if there is a person who is always in this viral infection a lot. Okay, um, and then if we ask right from all the kind of viruses, right from the bubonic plague, which we had in India way back in 1994, which we call it as a Gujarat plague, or the good old famine, the good old quarantine infections that took place in 1908. Uh, the one commonmost entity is these microbes are absolutely omnipresent. and they're absolutely mutating. So if you find one strain that is existing today, there is an immense scope of that particular strain mutating, and when we sequence that particular strain after a few days, you would definitely find at least one or two amino acid change in the evolving strain. So which means that you find a lot of mutations arising and falling. Okay, so having said that, if there is a mutation, even if there is a single most change in the amino acid, that would probably result in the non-synonymous mutation, and that might completely change at the way the protein is really looked into. So which means the proteins evolve and they also devolve. Okay. So next comes, yes, these are the molecules. These are all the biomolecules. So proteins as the largest biomolecules, they often tend to interact with each other. Okay, so which means you have a protein, that is sitting in the vicinity of the virion. That particular protein might be interacting with another protein sitting next to it, and they need to interact as well. Then only they cause the viral infection, and if they cause a viral infection, we'll be able to understand what are those antigenic determining sites or what are those epitope tags, and that's where we can probably target antibodies specific to those particular regions. Okay, so in this particular process, as the proteins evolve and devolve, the most important aspect is there might be some genes that are lost and found. Okay, so which means, so digging deeper into the kind of viruses, any kind of viruses, I'm not talking about the COVID. There might be genes that might be transferred from one organism to the other organism, and that transferee or that transferred gene might really aid in that particular viral infection. Okay, so there might be genes lost and found. So if you really look what systems aspects are important to understand all these kind of things. So we need some kind of analysis. We need to reach consensus. We need to understand the modeling concept. We need to take interpretation from the life sciences aspect. Of course, we need rigorous algorithms. We need databases. We need to visualize the things. So we need a complete overall picture of these kind of studies. So in a system, we always talk about a lot of integrated parts, integrated components that behave independently, and yet they also behave dependently on each other. So in case of virus, there are several proteins. A virus might have a single-stranded DNA or a single-stranded RNA or a double-stranded DNA or a double-stranded RNA. Ultimate goal is to understand how these particular proteins behave. Okay, so proteins as single singletons or single subunits of molecules, they have smaller uh, entities called amino acids, and these amino acids, they keep bind to different interfaces or sites. That's where the antibodies needs to be targeted. So from the systems point of view, all these smaller components come together, and we need to identify a better behavior on how these particular proteins come into picture. So if you ask me, the teaser here is what is COVID and what is unCOVID for layman's terms. So in case of COVID, COVID is pretty well you all pretty well know. It's a virus, um, and novel coronavirus is something that was first seen in November 2019 in Wuhan, and it has apparently mutated several times, and over 132 assemblies have already been uploaded in GenBank today. There are absolutely no complete assemblies. Um, I'm sorry, there's a small typo over there. It's not 1,000. There, it's 100. So over 100 complete assemblies are from China alone. So which means you just imagine from November 2019, the first what you call journal link that was updated in GenBank somewhere on November 29th till March 12th. So which means there are roughly about 94 days. So between by China alone, can you imagine how fast the systems biologists or the molecular biologists are working towards updating the sequences, which means they might have isolated the virus from a patient in the morning. They might have isolated the RNA. They have sent it for sequencing. The size is only small size of about 29.3 kb. It's not big. Okay, and they got the sequences. They assembled it quickly, and by night, they have uploaded it, and probably in GenBank or DNA Databank of Japan or all the publicly available sequences, they might have quickly uploaded it, and by next day, it was published. So we as scientists, need to evolve, so much faster, like what probably China has done. It's quite amazing. No one, I'm really in awe, about this. I mean, you have a sample that is ready, say 8 a.m. in the morning, and by next day 8 a.m. in the 8 a.m. Same time, you have your sequence uploaded in GenBank. So like which there are 100 isolates. I mean, 100 such assemblies. It's quite amazing. And if you observe the differences between these samples, and I'm sure there are a lot of changes, which which I'll probably describe you during the next few slides. So if you ask me what is uncovid like, so the uncovid thing here is the first 2 points are simply more towards COVID entity. The uncovid here is from India. The first outbreak took place sometime during February 23rd. That was the first case that we received, and only 2 of the sequences so far from Indian isolates. That's I would say pretty worrisome as a researcher, but from yesterday's webinar, I believe that the government is also working. I really don't want to open any Pandora box on this, but we are in a absolute sorry situation that we're not able to sequence at least as many as isolates to understand the behavior of these viruses. Okay, and the uncovid thing here is that many a time in case of the predecessor viruses like the Nipah or the Zika, okay, the MERS and other stuff, the samples were simplest blood. So which means out of about 3.2 GB haploid genome, you isolate about 30 kb of viral assembly, identify those particular sequences in the human blood sample, and then check what are those particular sequences, and then ask what are these particular sequences and how are these particular sequences different from other viruses.
Yeah, great.
Yeah.
You're there.
So the uncovid is that there are only 2 sequences from Indian isolates that are known.
Okay.
And the most important thing with uncovid is that there are only nasopharyngeal swab samples that are viably sequenced here in this case. Whereas in case of the predecessor sequences, we have the blood, the stools, and other samples also that was sequenced.
Mhm.
So here the most important thing is since it's a lung aided infection, the nasopharyngeal swab samples play a very important role here. So if you ask me, how different is this novel coronavirus from SARS human coronavirus, which is first most strain, in the family of the SARS, and of course, how is it different with MERS? So they all belong to beta coronaviridae, and over 70% of the 29.3 kb genome is non-structural, and the rest is about structural. So if you see the ORF1AB, which is the lengthiest most sequence, that is absolutely similar to SARS. That particular protein is quite similar to that of SARS human coronavirus sequence as well, and the rest of the sequences are structural. structural, which means there are four important structures which are abbreviated in the form of MENS. M for membrane, E for envelope, N is for nucleocapsid, and S is for the spike protein. Okay, so these four structural proteins are quite phenomenal, not only in novel coronavirus, but also from SARS coronavirus family members as well. So if you really go for a simplest blast, taking the entire novel coronavirus of Wuhan and blast it against the beta coronavirus members. With human coronavirus, that is SARS, it shows 99.180 with MERS. So a lot of discontent accumulates to about 66.3%. So which means there is a big structural deficit that is there with these particular sequences, which means there are many sequences, but there are very few structures. And if there are very few structures, identifying viable targets or epitope tags or the antigenic determining sites would also be a kind of a big challenge. Okay. So, so if you see, basically there are very important proteins apart from these MENS. MENS are nothing but those four proteins which I told you. Apart from this ORF1AB, there are also the non-structural proteins like NSP1 to 10. NSP10 plays a very, very important role because that is the particular protein which makes a replica of these particular protein sequences as far as the viral fragments are translated to the protein. Okay, so nevertheless, there are a few important reports, especially towards identification of epitopes. One Cell and Science paper last week revealed that the spike and nucleocapsid proteins have got inherent exposed amino acids that could be considered as viable targets. Okay. So, so one of these particular proteins is ORF1AB, which is well known. So ORF1AB is split into two important nucleotide contexts. Okay, and this is from our simplest bipep bipep predictions, which we call it as BPIPRED prediction. It's a server from CBS DTU.dk, which is a well-known university, the Technical University of Denmark. So you see the inherent structures. So wherever in the column where that is highlighted, E is exposed. Okay, B is the buried amino acids. So usually we must consider those particular amino acids that are exposed on the surface, so that antibodies can be absolutely targeted to those particular residues. Okay, when we say buried, so they are within, they are not really exposed. So the antibodies may not really be targeted to those buried residues. So interleaved, you may have to really focus antibody targets only on those exposed residues. And now you might ask me, what kind of exposed residues? So when you have a protein, has got several forms, structural forms, the helix, the beta pleated sheets, okay, the loops, okay, or the side chain sequences, and all those kind of stuff. So the most important thing is, if there is a helix, so helix would have an intertwined structure. So if there is an exposed amino acid, then targeting an antibody or an aptamer as a small molecule is absolutely very easy for them. Okay, so that's one of the reasons why these particular amino acids also are largely focused. Now coming to this highlighted point, as you could see here, the column that is highlighted to the left is nothing but the amino acid residues. So 1275, 1276, 1277, and in the down you have 1278, and so on and so forth. So the highlighted regions are tandem, which means they're next to next. So the amino acids D, I, T play a very important role because most of them are all the weak amino acid residues, and they are present at the exposed regions, albeit the fact that the exposed amino acids are surrounded by the buried amino acid residues. And if we have any of these particular sequences that are tandem, they play a very important role. So if you see here where my cursor is here, these regions could also be important, right, from 1181 to 1185. But you see the central amino acids, they're all buried. So you may not really target the antibodies over there. And by the way, these are all the scores across different different structures. Say for example, this particular structure is for for the LX. This is for the beta plated sheet, and so on and so forth. Okay, so these are the preliminary predictions that we have done, and as you could see, this is accession YP_009724 389. So likewise, so that was the case in case of ORF1AB. Then you might ask me, what is the case with the structural proteins as such? Okay, so there are some structural proteins, as you could see, the nucleocapsid protein. So the nucleocapsid proteins, you don't find any of them, tandem in amino acids. There is 252, 253, and then comes 89. So which means there is no 254th amino acid that polar comparatively, although these are all the exposed surfaces. Okay, so interestingly, you have 91, 92, 94, and 95, but that was not considered, and all of them have got about 16s. They're absolutely viable targets. Okay, so what amino acids needs to be targeted? So amino acid sequences came into picture. uh, uh, the most important stuff we often asked was, how different is this novel coronavirus from Indian isolate, creating with the other 132 odd complete assemblies. That was what we asked. Okay, so so as you could see here, the yellow shades, the viruses, two leaves. This is the simplest phylogenetic tree prediction that we have made from this. Okay, this clearly indicates that this is absolutely similar to the Wuhan. Okay, it is next leading to the Wuhan sequence, but it completely dissociates to that of the other bad SARS-like coronaviruses. Okay, so it is not really creating exactly here. There's a big gap. So the origin is somewhere here, the Wuhan, and they're absolutely similar. And if you see at the 4 o'clock position here, where the Wuhan is, and where the other viruses are present here, they're absolutely similar. But our viruses, our Indian isolates are present at the 3 o'clock position. Okay, and these are all the similar kind of sequences creating with all the 132 assemblies, and our phylogenetic tree is somewhere sitting somewhere here, where my cursor is. It's about 2ish, 2 p.m. Clock position. Okay, so this is where we can say that the novel coronaviruses played similarly, and yet differently. So in the meantime, there was a very interesting paper where Ahmad et al. This was published just 10 days ago. They identified you know uh, uh, the exact epitope tags, and identified the phylogenetic clades between all these MENS proteins, the structural proteins, the membrane, the envelope, the nucleocapsid, and the spike proteins. And they found an absolute percentage sequence similarity to that of SARS corona viruses as well. Okay, it shows quite similarity as you could see here. The entire green is entire protein with the MERS, okay, which is the MERS core. But you have a quite similarity with that of the human SARS coronaviruses. But only a small amino acid residues are um, are present, which are shown in red. That shows the novel coronavirus sequences. So this is a paper from Ahmed et al. So these provide us an absolute leads towards a systems understanding of the structural proteins. So in conclusion, when we say when we want to holy grail the sequences, when we wanted to identify the structural morphs for these particular sequences, if you ask me, as a systems biologist, I'm not a structural biologist though. I would like to suggest a structural biologist not to just target the structural MERS alone, but also target the non-structural proteins like the NSP1 to NSP10, primarily NSP9 and 10, which play a very important role here. Okay, there might be order of 1AB proteins which are quite similar as well, but there's a lot of variation seen in those particular sequences as well. And if we can identify those particular things, probably we can describe the function on how these particular structural MERS are really helping us understand, identify the viable targets, and as well as identify the viable antigenic determining sites, or even the epitope tags. Okay. So sometimes I always believe that we should not only focus on those particular proteins that are structural, but chop off what are not related, which means what are not really not structural, and then come to the final final pairs. So which means let's imagine you have a big basket of oranges. You wanted to keep the oranges that are absolutely clean. So you'll remove all those oranges that have been rotten. So simply remove those particular oranges that are rotten from the basket and keep those particular oranges that are alive and fresh. Okay. So that's certainly the need of that. So the challenges here is there's a need for bringing the challenge of understanding epitopes better, okay, or even the antigenic determining sites using bioinformatics and systems genomic approaches. Annotation and curation for better antibody targets, like what Ahmed et al have shown, or probably what we have really shown, where we are trying to ascertain the exposed and the buried amino acid residues and identify what are those polar amino acid residues that are absolutely making a myriad of it. Thanks to my postdoctoral mentor, we try to design different role. So together with Professor Giri, we are writing a grant proposal. They can give better specificity than antibodies. They're small molecules. They're cheaper, cost-effective, easily synthesized in the lab. The simplest MERS, simplest nucleotide MERS as well. They can be synthesized in the lab just like PCR oligos, albeit with a more optical density of 3 or 4. Okay, and they can be better targets. They can be RNA aptamers, DNA aptamers, or protein aptamers. But in this case, we're trying to work on the RNA aptamers, which we have already synthesized and which we are checking in the lab. And of course we need raw sequences from the genomes. Unfortunately, there are no raw sequences people are uploading to the GenBank, because we should always do the assembly first in-house in the lab by yourself. If you're doing that, that is a very important stuff. And for impatient, the assemblies are the best. Otherwise, if you think you're impatient, simply the assemblies, the 132 assemblies that are published online, they are the best. So I think I thought I should also discuss these kind of things for a good Q&A session. I think COVID-19 is really taking us towards a better understanding, towards towards making towards making not only better targets, better vaccine targets, or better identifiable targets, but also it is also allowing us to understand uh, sorry so where the collaborative efforts are playing a very important role. So I think for this one of the important questions that we need to ask is, where is COVID-19 taking us as researchers? Okay, I think for that uh, um, I just leave it to your discretion. So we need collaborative efforts and all those kind of stuff. And the next question, probably what I would probably ask you to ask is, what are your thoughts on the nature of the novel coronaviruses and the claims of its origins from the lab or transmission from animals to humans? There are three reports as of now where zoonosis is not ruled out. So many of these coronaviruses are infected from animals to humans as well. In case of the dogs, there was one report. In case of one cow, there was one report. So there might be the case as well. The other question that you might ask is, what advice we need to give as a researcher for someone treated or affected with virus or from virus? Okay, there's a small or subtle difference with that. So probably we need to ask such questions questions as such. And how prepared are we in India at the diagnostic front? Probably we are absolutely prepared. Thanks to the wonderful diagnostic centers coming up with PCR kits when compared to the rapid diagnostic kits. There are also some lateral flow assays that people have started developing it. And I think the next week we'll see some of the sensitive and specific tests that are coming into the market very soon. And the most important question that we need to ask is, as a bioinformaticist or as a systems biologist, as a genomicist, as a biologist, uh, how difficult is to assemble these genomes? So if we have a raw sequence, when China could sequence one isolate every day and publish it the following day in GenBank, why not we? Why not we as biologists? We have even the USB-based sequences like the Oxford Nanopore technology sequences. We need to take up these particular challenges. We need to bring up this attention to our bosses, to our institutes, to different stakeholders, and say, hey, look, I have the capabilities to assemble the genomes. I'm a bioinformaticist, I'm a systems biologist, I need to do this. I can bring up that kind of conference to you people. So this is what we probably we need to ask. And I think this is certainly the need of the hour. And we need to bring up that competing levels and confidence levels to the fullest, and ensure that we reach the pinnacle of success. And I think there's absolutely the need out there where clinicians, geneticists, systems bioinformaticists, molecular biologists, virologists, IT people, the people who are into artificial intelligence, everyone come together. And I think COVID has really taught us new lessons, not to be just at the level of precautionary measures, but also rich in vivid thoughts. This is what I can really say. And I think the onus is on us as researchers. We need to educate masses. We need to educate our family members, and we need to educate our brethren, and we need to ensure that it's stopped only at this particular level, but we need to take it to a larger and bigger front. So this is what I really sort of saying. I should really thank really for this wonderful time, and I really look forward for some kind of questions. I have a small 2 or 3 graduate students. So if anyone is really interested to work virtually with me, I'll be happy to take. Yeah, thank you so much.
Okay. Thank you so much, Prash. It was very much insightful.
Hello.
Can you
I
hear me?
hope I'm not alone.
Can you hear me? Yeah, yeah. I have unmuted myself. Can you hear me? Hello. Okay.
Hello.
Yeah, I can hear you. Can you hear me? I hope everybody is able to hear me. Okay. So we are just continuing
Yeah,
with.
yeah, yeah. Thank you. Yeah.
Great, great. So, okay, so if when you are able to hear me, it's good. So, thank you so much, Prash. It was really insightful, and
Very good.
I really liked the questions and your opinions. I'm not going to comment too much on the.
I thought I was just alone. I'm sorry. No, I thought I was just lone speaker. Yeah.
No, no, no,
Thank
no,
you.
no.
Yeah.
I'm there. I'm there.
Yeah.
So
Please.
the only thing is that
Yeah, 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 yeah.
yeah.
Please, yeah.
So what I thought was you rightly put forward, because in order to fight this pandemic, a collaborative effort is very much required. That is something that everybody should know about it. Individually working is not going to make a sensible answers for the questions that have been raised. So a collaborative effort is very much mandatory. And the second thing that you also raised was very true. When others have done in just a matter of 1 or 2 days, why we cannot do when there are a lot of experts available? Of course, we are in the lockdown position. There are limitations, but I can see that there are many residential campuses where people are still actively working in many institutes across the country. So why not we? That's a good phrase that we can ask to everybody, and we have to see how they can come forward and work collaboratively to come up with something that is addressable. And the other point also which you have raised is we are just focusing only on publication. I really agree with you, because all these preprints websites are now filled with COVID publications. Now the question is which one I should read, which one I should look at. The ones which is being widely shared in Facebook and LinkedIn and in other social media. So we are a bit biased on which one to pick the right one. And I saw that in GitHub there are machine learning and like language processing algorithms trying to find out the best pick from the lot collection of publications. So we have to ensure that I really like that what you have put up. Rather than publication, let us nail down and try to really understand what is the actual reason behind all these problem statement, and then come up with your assumptions or your findings or your hypothesis or even your perspectives. Sorry to say these words, because in COVID-19 I cannot use a confirmatory word, because everybody is trying their best to come out of to answer those questions. So really thank you so much, Prash, for your insights, and I hope there are more. There are about 55 participants with the Google Meet as well as in the YouTube. They might have got a benefit from your findings on your pilot study. Now we have around 14 questions in Mendy.com, and I hope everybody is able to see the questions on the screen. So the first question: are there any mutations reported in COVID-19 up till date? I hope you have already mentioned about two, but over to you, Prash. You are the best person to answer this question. Prash, there is a delay from your side. I hope you have to switch off your webcam. We are not able to hear you. There's a bandwidth issue from your side, I think. You are unmuted, but not able to hear you. You can try switching off your webcam. That would be the best. I think now we are able to hear you. You can try speaking.
Yeah.
Okay, great. Uh, Prash, it's still not.
Yeah. 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 Thank you.
Yeah.
From my understanding, there are mutations, but here, identifying mutations is not the key
Sir,
story
we
here.
cannot understand you, sir. Your voice is
Uh,
breaking.
whether or not those mutations have intended targets.
Prash, your voice is breaking.
Yeah.
Yeah.
One minute.
Now you can try.
Yeah.
Yeah.
1 minute.
Yeah.
Yeah. I think I'll do that.
Yeah. I think your webcam is on. That's why. Yeah.
Yes. 1 minute.
Sure. No problem. Yeah. Everywhere there is a problem with internet speed and bandwidth, so it's understandable. No problem.
1 minute.
Sure.
Sir, because quarantine, sorry, lockdown is going on, so
Everybody is
by
working
the
at home.
government,
Yeah. Yeah.
everybody is using.
Yeah. Yeah. Go ahead, Prash.
Why?
Now it is better.
Oh,
Now it is better. If your webcam is off, it is better.
this I do this. I don't know why it's not working. 1 minute.
Now we can hear you properly. No issues. Yeah.
Okay. Yeah. Even your voice, I don't know why it's getting delayed. Giri, I think Giri, I think but I'll just go ahead with my point.
Yeah.
Um, what I video here.
Yeah.
Yes. Yes. Now I think it stopped. Can you see it's stopped now? 1 minute.
We are able to hear you now.
Is it audible now?
Yeah. Yeah. Yeah.
Yeah. Yeah. So
Yes, sir.
yeah, yeah, yeah. I also find a problem in my house. Yeah, yeah. Reported in COVID-19, no doubt about that. As we so there are mutations reported here. As long as we find intended targets, this may the structural sites play a very important role for for antibodies. There are mutations that are across different both non-structural and structural proteins as well. Specifying mutations, we just need to ensure what are those or for that particular mutable region where we are. I think there's a big delay between my voice and my. I'm sorry for this. Using the bandwidth that we have on aged 12 years and my wife, we have got different gadgets where this internet is being used. That's the reason why it's delayed.
No
Okay, I'm sorry
problem.
for
You're
this.
good. No problem. Okay. So he has already made his points clear on the mutations. Is there any further points you wanted to address, Prash, or I can go to the next question?
Yeah, you can go to the next question.
Sure.
Yeah, please.
Thank you. So the next question is N95 mass pore size ranges from zero point 0.3 to zero point 0.7 micron. Yeah. But coronavirus size is 0.12. Yeah, 0.1214. Yeah. And how we believe the N95 mass for protection. Okay. Any comments from your side?
Yeah, you know, yeah, I'm sorry, I'm not an expert in this, but I have my own personal take on this.
Yeah.
It's not about the mask porosity or the mask size here that matters, or even the viral micron size. Now, what happens is when someone coughs, the virions are spread across different dimensions.
Mm-hmm.
Okay, and you'll not have the same micron size of that particular virion contagion, affecting the the possible or the plausible infected person. So
Okay.
it would definitely change in real time. So So this is what I could think of in that. I'm not really probably a good person to answer this particular thing. Maybe Giri, if you have any point on this, I'll be happy.
You're right. I really don't have an understanding here because until this. Please. Sorry to interrupt. Please, madam. Can I answer this question? Please. With your permission. Of course, it is an interactive session. Anybody can. Virus that is present in the aerosol is in the droplets, water droplets, with the saliva droplets. Uh-huh. And the mask, this mask or even mask with a lower configuration is good enough to hold back those aerosols. Virus, in any case, whatever size it is, if it is present as a single virus, it will get dried up very fast in the air and it won't be contagious anymore. So you basically are protecting yourself from coming in direct contact with the aerosols, that is the droplets of saliva which contain the virus. Mm-hmm. The size of the droplet matter, not the size of the virus. Or bacteria, okay. Or whatever contagious organism is there.
Right.
Oh, great,
Yeah.
great. Okay.
And the size of the droplet is approximately less than 100 microns, so definitely it can protect, sir.
Yeah, sure. Yeah. Thank you so much, Dr. Vandana. That was a really nice answer.
You're most welcome.
Yeah. I'm going to the next question. That is, do we have any details on the variants in India? Of course, there were two of them were sequenced.
I tried to.
Yeah.
Yeah. Two sequences available, and from our understanding, with interaction with NIV directors and NIV scientists and IGIB scientists, we're going to sequence a few more isolates, by the end of this week, and hopefully we are to get the assemblies, sometime by Monday next.
Okay.
But my point is, more beyond that. It's not about the assemblies. It's about the raw rates. The raw FASTQ rates or the raw FAST5 rates in case of OMT sequences. Mm-hmm. So we need the raw rates so that we can analyze or we can assemble the genomes ourselves, because there's a lot of crap that's being published these days, and not many sequences that are being assembled is really the cup of tea from a bioinformaticist point of view. So as a systems biologist or as a bioinformaticist, we'll be very much contented and happy if we assemble the sequences ourselves. So no one is trying to publish the raw rates online. So it's only the assembled rates. And I think I wouldn't really trust the assembled rates alone. As I told you, if you are impatient, simply trust the assemblies and go ahead. So having said that, specific variants from these two sequences that are published doesn't make any sense. We need at least 20 or 30 sequencing sequences where we have isolates from different different population, and then we can identify the variants relevant to India.
Okay.
Yeah. But as of now, there's no big difference between our isolate and the Wuhan, excepting a few bases. Yeah.
Okay. Thank you so much. And the next question, what factors do you think it's responsible for the emergence of viral variants?
Yeah, I think one of the important aspects is the dietary lifestyle. That is absolutely very important. So I think if the question is related to Indian condition, I think we're absolutely immune because we take a lot of spiced food. We take a lot of curcumin. We take a lot of ginger. We take a lot of garlic, and we take off a lot of hot beverages and all this kind of stuff.
Yeah.
Although there is no research prevailing prevailing with respect to the exact or impromptu emergence of the viral variants. A certain point is related to the dietary lifestyle from the ergonomic front, and from the scientific point, I think I would really correlate to the horizontal gene transfer of some of the sequences that might have emerged if at all, if people have really taken some non-vegetarian food, but those particular reports were also not justified in
Yeah.
case of the Wuhan Chinese sequences. Whereas in Europe, uh, the emergence of viral variants, in case of Europe or even Americas, both South America and North America, because of the temperatures, right from 3 degrees to 16 degrees, because the viruses, the RNA in viruses are absolutely labile. They tend to replicate very faster. They tend to live shorter, but they're going to make the infection very quickly
Okay.
as soon as it emerges into the viral assembly, or as soon as it emerges into the patient or into the host. The emergence of viral variants could also be attributed to the temperature as well, kind of conditions that these particular um, the host is associated with. So for this, I forgot to answer one question from my presentation. So we are also trying to develop a COPASI model. A COPASI is a complex pathway simulatory model where we use a lot of differential equations, set the temperature. We try to give an artificial virus. We give a sequence. We ask how this particular viral sequence replicates if the temperature is increased, say from 28 to 34 degrees Celsius, and then we
Okay.
increase it to 36 degrees Celsius. Then we increase to 40 degrees Celsius. So these are all just heuristics. This is what we call it as a complex pathway simulation heuristics. Yeah, this is also being
Okay.
done by us. Yeah, as of now.
Oh, great. Since you have mentioned about our Indian, of course, food style, very true, because that's where I could see Ministry of Ayush also was requesting for trying to increase your immunity rather than people shouldn't take the message in the other way around. It is just to fight against COVID. It's just all these our food habits are going to help us to increase the immunity. Am I right?
Right, absolutely. So that certainly plays a very very important role.
Yeah.
Needless to mention that that's absolutely well inserted, Giri.
Yeah.
And this is certainly one of the reasons, as you could see, none of those particular patients were bona fide viral infected patients,
Uh-huh.
from India who has had no travel history at all. There are very few. There are one or two, where you can count. So all the contagion has really spread from those who have really had travel history, or who are above 60 degrees, or who have had some kind of autoimmune diseases like lupus, or who are really smokers, or who have had asthma, who have had some breathing problems, who has had some problems with asphyxiating inheritance from their parents, or all this kind of whisky, but not relatively a healthy commoner
Mm-hmm.
who is really associated with this viral infection. Yeah.
Okay. On this context, I have another question from one of the participant. What about asymptomatic variant?
Yeah. So the asymptomatic variants are also being studied. That's where we need to ask. So the very first question, we need to segregate from asymptomatic and symptomatic variants is whether or not these variants are really present at the exposed sites of the structural protein. So let's assume that you would find a particular variant that is absolutely asymptomatic.
Mm-hmm.
Okay, and that particular variant is not really causing the disease, and the patient is absolutely healthy, and the variant is really sitting in the structural morph of a protein, where you find that particular variant to be present at the exposed surface.
Okay.
And now, likewise, you have a symptomatic variant from the deceased patient, from the same novel coronavirus infected patient,
Mm.
you find the same variant probably tending to be symptomatic. And now comes your question whether or not that asymptomatic variant is surrounded by any buried residues, or in that particular uh, sequence, or in that particular isolate, whether or not the amino acid change has really given a complete change in the structural morph of that particular sequence. So if there is a E, which is an exposed amino acid residue, which is a
Okay.
variant which we call, it is surrounded by 2 or 3 buried amino acid residues, then that is what we call it as asymptomatic variant. Whereas in a symptomatic variant, you have an exposed variant,
Okay.
which is E, that is also surrounded by few other exposed amino acid variants, and then followed by the buried amino acid residues. So that
Okay.
is where the difference between asymptomatic and symptomatic variants come into picture. So in a nutshell, from the sequence perspective, in case of asymptomatic variants, E is surrounded by 1 or 2 Bs, which means B is buried, E is
Mm-hmm.
exposed amino acid residue.
Mm-hmm.
Whereas in symptomatic variants, you have E that is surrounded by another 1 or 2 E's, which are
Okay.
again exposed amino acid
Exposed ones.
residues.
Okay.
So there is a slightly different from the structural perspective.
Okay. Hope we answered the question.
Is that a mutation?
Can you please repeat it? Hello?
Is that a mutation?
Prashi,
Yes.
she's, yeah, okay. Oh, you heard it. Okay.
Yeah, Nancy, that's where it's not still a mutation. It's still we are trying to ask whether the same variant is seen in asymptomatic patients and as well as symptomatic patients. Okay, so which means I'll just give you an example. Let's take the protein sequence, say MGKST. K-lysine is a variant which is present in asymptomatic patient.
Mm-hmm.
Okay, and the same K is also seen in asymptomatic patient as well. Now, in this particular sequence, since there is MGKST, in symptomatic patient, there might be MGKKT or MGKLT. So, which means the K is surrounded by another exposed amino acid residue or another mutated amino acid residue, which is L. So the variant in question doesn't vary. The variant in question here is we're only asking what is that particular variant. So in this particular case, the variant is only K. It is not L.
Mm.
Okay, so we are only looking at those variants surrounded by that particular variant in question. So the variant in question here is the K. So we are not asking whether or not that K is mutated or not. The K wouldn't really mutate. So even if it were mutated, then that is out of context in calling it as asymptomatic variant or symptomatic variant. Okay. So in
Okay.
asymptomatic variant and symptomatic variant, the amino acid wouldn't change. It is the same amino acid per se. Okay. Yeah.
Okay. Okay. Thank you.
Thank you, sir.
Now going to the next question, what basic equipment would we require at the university level to carry out research? Of course, I understand it is related to COVID-19. That's a topic anyway. Sharing any research with fellow scientists. But before answering this question, Prash, I want to just have an understanding. Anything that you're going for vaccine related or drug related with this particular COVID, I think you need a BSL-3 facility, right? That is a certification required.
Exactly. Yeah.
Yeah.
That is
Sorry
certainly
to interrupt, but
certainly.
I was the one who asked this question, so I just want to give you the information that
Please.
this is assuming that BSL-3 is available in the university.
Ah, okay. Okay.
Yeah.
Thank you,
Yeah.
Vedant.
I
Yeah.
think that's certainly a very good question, Vedant. But I think as Giri points, there are very few labs or universities. I mean, especially the medical colleges, have got this BSL-3. Not all, and not all are allowed to have this BSL-3
Yes.
facility
Yes.
because you've got various safety concerns and
Yeah.
the ethical issues. But the academic front, that's certainly the need of the hour. I think I certainly mentioned it, in one of my slides, that we from academia need to have at least the BSL2 license in the near future in order to carry out these this kind of research. At least the vaccine testing or the drug testing or any other things can be done probably in certain industries or in certain medical colleges. But at least from the serological point of view, or simplest antigen antibody test, or simplest epitope tags, or simplest rapid tests, or simplest PCR based kits. I think I'm sure that Government of India or the Medical Council of India would also suggest that we should have at least BSL2 pens,
Okay.
at least from the future. I think this is what at least we should probably, and not only that, not just having the license is important, but having a trained manpower.
Yes.
This is very, very, very much important. Maybe if you're not trained properly in this particular direction, then we're at a huge loss.
Yes.
Yeah.
You're right. But as he has already mentioned undergrad and postgrad level, is there any other comments from your side?
I mean, I would also rate it to that level only. I wouldn't probably suggest having a BSL2 at the undergraduate level. At least at the postgraduate level, we should do that.
Okay.
I don't think we are. To be very honest, I'm not trying to be pessimistic. I apologize, but I think our schooling, our STEM curriculum is not really prepared
Yep.
to do research in virology at the undergraduate level.
You're right.
I need to be very open on this. I'm really sorry for this, but I'm sure we'll be prepared. It's not that that's the end of the day. Probably we'll certainly be prepared 2 or 3 years down the line.
Yeah, you put it forward right way, Prash. There is no need to feel sorry at this particular era. People should know that we do not know things, and we should admit and we should upskill ourselves, and we need to know what is required for tomorrow. And there is no need to feel sorry, and we have already time has passed to realize that we are not upskilled yet to face some challenges in researches. It's a fact actually. Okay. Um Thank you. And going to, okay. So the question here is, so Prash is going to answer the question. So the question here is, can membrane mimicking nanodecoys be used for viral sequestration?
Yeah, I'm sorry, I'm not an expert in this. Maybe Giri, if you could answer.
I already told them I really didn't understand
Yeah.
the word sequestration, so I already apologize for that. I don't know. So
Great,
probably
great.
we have to get them back. Otherwise, there is one of the attendee, Dr. Selva Kumar, is trying to answer. So please try to answer if you know.
Sequestration, the minimizing this viral impact.
Okay, sequestration is to minimize the viral impact. That is the word meaning. Okay. Okay. So we will try to answer this probably via email after just doing some little bit of research on this. Is okay. Then three variants are already reported. Should we expect more?
Yeah, certainly. I mean, this is certainly going to be mutated. I think that's really a good question. I think, we are going to have at least we're going to see a complete overall of the mutated genomes, during the next 3 or 4 weeks. Let's start this way. Let's take the predecessor viruses, that we have had, right? From MERS, Ebola, SARS, the SARS human coronavirus, Nipah, Zika, and all those kind of things. There has been inherent mutations that have been reported right from day one till the end of the epidemic stages.
Okay.
But considering the fact that this is pandemic, and whether or not there is an epigenetic influence is not yet known, we might definitely expect a lot of new variants coming up. But as I tell you again and again, it's not the number of variants that we need to probably understand whether or not these variants that we intend to see, where we intend to target antibodies, could they be the viable targets or not? That is the question that we need to ask, whether or not these variants could really be translated at the helm of understanding better antibody targets. That is the most important question that we need to put it.
Okay.
So this is my take.
Okay, thank you. There was one more question following up this one, that there are like herd immunity. Of course, we are discussing a lot about herd immunity, but in Japan as well as in China, the people who have recovered from the infected coronavirus, they are also again getting infected again. So what is your take on that?
Yeah, I think that is expected in terms of any contagion, and if I'm not wrong, I think I hope the questioner is not relating to the question where the same patient is infected again and again. I hope that is not the point.
The point is same. The patient is getting affected again. Yes.
Yeah. If that were the case, maybe the interferons in that particular patients are really suppressed in such a way that the kind of drugs, the proprietary drugs that he has been advised with, are not really making a merit in his body. That is the simplest scientific take from that.
Okay.
So that is number one. Number two, the patient, he or she might have been might be associated with this suppressed immunity because he or she might be associated with one or two more autoimmune diseases inherent to his body. So if you see, I think there are only 3 cases that have been reported, and that is a rarity, and that is also a rarity, for COVID science. You can't expect a kind of a patient, being affected again and again. It's just like the kind of drug intake that particular patient is posed with. I'll just try to correlate this example with our human targets. I'm sure that we have had the BCG vaccines right from early days until 2003. All the vaccines that were prepared, they were all the DNA-based vaccines.
Yeah.
Okay, and all the DNA-based vaccines are absolutely inherent, and they play a very protective care and gives immunity, for different diseases. Whereas Comet 2004, all the vaccines that were prepared, they were all the protein-based vaccines. The protein-based vaccines are not that sensitive. They're not that immune when compared to the DNA-based vaccines, albeit the fact that the protein-based vaccines are pretty cheaper. The DNA vaccines were very robust, very duty-bound, very target-bound, and it brings in a lot of antibodies. It brings a lot of interferons inside the body, and it gives a kind gesture that the antibodies are produced in tandem, right from day one of vaccination. And most importantly, at the 3rd day of the vaccine, the patient would be getting a small fever, and there would be a small relapse as well. Whereas in the protein-based vaccines, you see seldom the children get any fever these days.
Yeah.
Okay, that's because the proteins are really depleted inside the body and inside the veins. They're absolutely not transmitted, or they might be coming out in the form of excretion or whatever we could think of.
Okay.
So the subtle answer, the bottom line here is, it all depends on how good the vaccines are really bound or duty bound to that patient, and whether or not that particular patient has got one or more systemic infections or systemic autoimmune diseases associated with him.
Okay.
That's my point.
Okay. Thank you. There is another question. Have you inspected SARS-CoV-2 genome, especially spike?
Yes. We have done, the four, not just the spike. Of course, spike protein is a main target. But as a systems biologist, in one of those particular slides, our take is not to remove the rotten oranges, from the big basket. So what we're trying to do is we're trying to remove all those unnecessary or not so important proteins that are really not making any sense towards the antibody targets or towards the interferon targets or the aptamers that we are trying to design. In case of spike protein, the challenge here was at least from the Wuhan perspective, the structure is well deciphered. There are certain exposed amino acid residues in the spike protein. It is absolutely something similar to that of the SARS human corona virus, the erstwhile SARS human coronavirus.
Okay.
And yet, the same targets, the same antibody targets, or the same targets could not be viably used for this particular novel coronavirus. So that's a kind of a big mystery. So which means not just the spike protein, but also the other four structural proteins like the MENS membrane envelope, and the nucleocapsid protein also plays a very important role. But apart from that, to me, the most important protein that would really necessitate the systemic function behind this viral infection is the NSP proteins, NSP 1, 2, 3, and 10. So the spike, interestingly, would definitely interact. Simply go for a simplest viral protein interaction database and search the spike protein. It often interacts with some other proteins as well. Yeah. So now you ask a question: what if you chop off that particular protein from the virus? What happens to the virus? How good the virus would really interact with other proteins in causing the viral infection? So the spike protein is certainly a good cup of tea, but the only thing is it's not really the only target. Okay? It shows the quite similarity with the erstwhile SARS human coronavirus sequence, but no such justification is really made towards identifying viable targets. Yeah.
Okay. Thank you.
Yeah.
The next question is, any idea on SARS-CoV-1 genome and its animal to human transmission?
Yes, the beta SARS members have been considered to be more or less zoonotic. But the last several years of SARS research, the only thing that is often seen is with respect to the typical zoonotic states of these particular viruses prevailing. Okay. I don't think this is a kind of a matter or a topic of interest only for SARS alone, but it's also for other viruses as well, not just this. I think that was what I wanted to say. Yeah.
Okay. Another question: can you explain a little bit about genome level variations of SARS, MERS, SARS-CoV-2?
Yes, I think that was what I described in a few slides. Also, I didn't go into detail. If you really see this novel coronavirus is 99.1% minimum similarity with SARS human coronavirus, and about 66.1 or 3% discontiguous with the MERS. And if you see the commonalities here, the ORF1AB, right from the first 70% of the entire genome assembled sequences, we have got a big open reading frame called 1AB, which harbors a big glycoprotein, which is a big polyprotein, which harbors a lot of small proteins together, and these proteins together play a very important role. But these are those particular proteins that have a lot of inherent exposed amino acid residues to the brain.
Okay.
Whereas the structural proteins, the MENS and also the other proteins that we are always talking about, these particular proteins have less exposed surfaces, more buried structures, and lesser targets for these particular for the proteins.
Okay.
So having said this, if you ask what is the commonality between these three, I would only say that maybe structurally they're absolutely trying to be a bit similar. But if you really understand the structural morphs, or if you understand what are those exposed residues inherent to these particular three structural proteins of three different viruses, they're absolutely different. So which means identifying viable targets, or identifying better antibody targets, or identifying the antigenic determining sites in those particular protein sequences also play a very important role. Okay.
Okay. So yeah, we will go to the next question. That is, researchers are focusing on spike protein, but is there any scientific evidence for the immunogenicity of envelope protein in SARS-CoV-2? Oh, just a minute. I think there is again a connectivity. Okay, trying to reconnect. Just a minute. All technical glitches are coming up. Sorry about that. I know it's lunchtime and we are still holding back. Sorry for this. Hi, Prash.
Yeah, sorry, Gary.
No
so
problem.
No. So this is what I thought from the genomic perspective. There is certainly a big variability. There is a big discontiguity. But one of the important questions that we could say is that genomic similarity is not the question here. What matters is whether or not at the structural side, especially at the intended targets, are there any exposed amino acid residues that play a very important role?
Okay.
That is certainly the need of that. Yeah.
Okay. Thank you. The next question, researchers are focusing on spike proteins, but is there any scientific evidence for the immunogenicity of envelope proteins in SARS-CoV-2?
Yeah, I think there are a lot of scientific studies. I think the recent Cell paper also states that even there are intended drug targets as well. People have done it.
Yeah.
People have done even the proprietary drugs, even the repurposed drugs have also been tested both at the in silico levels, and as well as at the in vitro level based studies. There are a lot of drugs that have been poised to treat not just the envelope proteins or the spike protein targets, but also some other regions as well. So I think there have been extensive studies. I don't think we need to worry about that. Yeah, I think the only target that we need to really focus is what are those regions within them that are permeable for the exposed sites, so that better antigenic determining sites and antibodies can be targeted against them.
Okay.
Yeah.
Okay. Great.
Yeah.
Now the next one is, in about 17 years, if mutations in virus can make them to jump the human barrier, why can't it be reversed also, if it is natural?
Yeah. Something. I think that's that's a good question. I'll tell you a rationale for asking these such kind of questions. I appreciate the questioner for asking this. I'll tell you the sad story of we scientists. We as researchers or scientists, we are so-called atheists. We are not supposed to believe in God actually. But in principle, people like me who pray to the God, who believe that yes, we are good. We also do some puja stuff. We do these kind of things. We also believe in God. We also believe in something called supernatural powers. To the 30th thing. At least an iota of thing. At least 1 or 2%.
Yeah.
And that 1% of thought should really make us permeable or amenable in bringing up a thought that yes, we should not think of those kind of stuff.
Yeah.
Probably one answer to this your question is yes, that maybe 17-20 years down the line, if this can happen, it can also be reversed as well. But in order to stop this particular thing, the only solution, the only answer lies only in the way we live. Or in the way we ensure that our brethren lives, or in the way we ensure that our society lives.
Yes.
In one of my blogs, I raised a very important point. Why is that the trash lifter on the roadside wouldn't get any disease at all? You see, take the trash lifters in India. They're absolutely prone. Their body is immune to various conditions, various diseases.
Yeah.
They take all the unnecessary stuff from the trash. They don't even probably wear the gloves as well. But why is it no COVID, no other diseases are associated with them?
Yeah.
Whereas a person who drives a scooter, which is some 15-20 meters away from the trash, gets all the diseases, from right from A to Z.
Yeah.
So it can be reversed. Certainly, it can be reversed. But I really associate your question, more towards the lifestyle, towards the way we live, or towards the kind of scientific partiality of how we could live, so that we can evade these kind of points. Yeah.
Got it.
Yeah.
Thank you.
Yeah.
The next one is, what changes are there between SARS-CoV-1, CoV-2 that makes its rate of transmission high?
Yes, I think probably virologists can better answer this, but
true.
I'll probably put it this way. In any case, these are all single-stranded RNA viruses, which means RNA is absolutely labile. Okay, it's absolutely short-lived. So what it thinks is, it will, disintegrate very fast. And as soon as it disintegrates very fast, it wants to attack as many as different hosts and different systemic cells in the body. And then it causes, or it spreads the virus all through the body. So that is the commonmost thing across all the three, virus family members. But one thing that is not common is the mode of infection. Okay, so the mode of infection here is this novel coronavirus is more systemic only at the nasopharyngeal cavity. It causes a lot of inherent diseases towards lungs, or towards breathing, towards asphyxiation, towards COPD and all those kind of stuff. So maybe it is giving us new lessons. It is trying to ask us some kind of questions. Hey, look, I'm a virus. I'm going to spread even to those particular people who are really smokers, or who are even not even smokers, or who are passive smokers. And now I really want you to put a question that, please stop me from infection. So far there are a lot of studies only on probably COPD or lung-related diseases, or what you call bronchitis and all those kind of things. But maybe the researchers could also look into a specialized thought whether or not the person is really has got some insights of COVID-associated things.
Okay.
So we have asked one question, but the answer has really led towards a different thing here, because we are trying to ask how different are viruses, and yet they are absolutely similar across different phases of systemic infection. Yeah.
Okay. The next question, probably we will not answer, but I wanted to read the question. No controversy. Personally, what do you feel? Is SARS-CoV-2 bioengineered? Yes or no? I don't think we are not focusing on that, which I already had a disclaimer that no socio-economic discussions anyway. So I'm going to the next question. How PG chemistry students can help to fight against COVID-19? What do you suggest for all students? Do you have any comments, or should I take it up?
Giri, would like to go first?
Yeah. So, of course, you guys are the bread and butter. You can only make something new and something derivative. So, repurposing, of course, that is a different side people are looking at. But if you have some really good ideation in your mind that you can come up with a medicinal chemistry perspective, not a normal chemistry. Of course, chemistry is the parent area, but you have to more focus on medicinal chemistry to understand the pharmacological perspectives of the mode of action mechanisms. So that is where very important. But if you have any other comments, please, someone has unmuted. Yeah, Himanshu, yeah, you can tell.
Sir, computational chemistry can help them to search active molecules from existing molecules. Our university, Maharaja Sayaji Rao Gaikwad University, students of the PhD students are doing that.
Okay. So I agree with you, computational chemistry, but computational chemistry is too early from getting the drug in the market. So ultimately, whatever we do computationally, the chemists have to make the compound on bench, and that has to be tested, and that has to go for the humanity. So on the research perspective for a PG and a UG, of course, as you rightly said, computational chemistry can really help them to do preliminary analysis, hit identification, lead generation, but ensure whatever analysis you are doing that has a scale up capability and that can also be transferred to the lab work. That is very important. Yeah. So Prash, if you have anything to add up, please.
Yeah. I agree with your points, Giri. But I think this is a challenge for each one of us to ask questions in these platforms. This is how it is. I mean, if at all, if you're asked to do any service for COVID science, the best thing is to not to stop asking questions.
Yes.
So we should keep asking a lot of questions, irrespective of science that you're trying to do. So COVID has really opened us a lot of different platforms, not just biologists, but also people from AI, people from IT, people from physics, people from manufacturing, people from other spheres. We need to join hands. We need to ask the kind of questions that are not asked before.
Yes.
That is what you could do.
You're right.
And as an undergraduate and as a PG student, probably you can think of an entrepreneurial science as a discipline. That is very, very, very important. We have only one thing in our mind, that yes, BSc after BSc, go for MSc, maybe everyone is doing PhD. We should also do PhD, then go for postdoc, then go for or land up in academic front.
Yeah.
And very few probably go to industry as again for job, because they want more money. That's a different side of story.
Yeah.
But no one would really think of becoming an entrepreneur himself or herself.
Yeah.
That's a pitiable situation.
Yeah.
So maybe post COVID, I think this is the kind of question probably we need to ask, whether we are trying to come up as entrepreneurs. Maybe who knows, in the simplest an engineering graduate can automatically develop a sewing kit where if a cloth is dropped into a machine, it will automatically develop a mask.
Yeah.
It will automatically convert into a mask. That's his manufacturing story.
Yes.
That you can think on.
Yes.
Maybe as we can think of a small serum-based antibody kit. Now, if we are really good in biology, or if we are really good at algorithms, maybe to detect better interfaces, better exposed amino acid residues, a bit of tags. That is where we can think of, and try to convert this knowledge to the translational level, not just publish. Publish and perish. Maybe stick. Maybe it's only at the academic stuff.
Yeah.
But I would try to slightly put it this way, that it's not publish or perish, it's perform or perish.
Yes.
That is very, very important.
Very
that is
important.
certainly, we need to, COVID has really taken us, very strict challenges. We need to perform. We need to identify what is best in us and try to convert that into translational knowledge and become, good entrepreneurs. And the best example here is, Giri himself. Giri, with his vast experience, he has wonderfully, converted himself as a good entrepreneur, and he's doing in a human service, these days in organizing, such wonderful webinars.
No problem.
Yeah.
Thank you so much. So to add up there, there is a company called Post Era. That company is a very, 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 I should say the startup very recently started, only after the COVID has started. So, and they have scaled up like anything. They have investors. They are going forward to synthesizing and going for testing. That is a startup just came within this 3 months of COVID time. So as you rightly said, there are possibilities of more new startups that spin-off can happen based on the research findings and questions. Great. So the next question, do coronavirus fly in the air?
Yeah, I think that is also a good question. Going around. So it's not completely community driven, or it's not completely airborne. But one thing I really ask here is, interestingly, if you might have observed, from Wuhan, from Europe, or from US, the virus has spread only during winters, and not when drastic aerosols are around, and not when many pollutants are around, and not when there is too much of snow, or too much of cold climatic conditions. So it only has spread between temperatures 3 and probably 18 or 19. Be it Wuhan, be it South Korea, be it in some phases of Australia as well, because Australia has also gone to winter by now, and some aspects of some states of US as well. You see, in California, you don't see that much as you see spread. So I don't think it's completely airborne. It all depends on the kind of mild pollutants and aerosols present in the environment.
Okay. Okay. The next question, I don't know whether we can take it up. Many problems exist by untrained employees that they have knowledge in virology. How to rectify it? Problem exists with equipments like ventilators. Moreover, government hospitals don't have virology based safety cabinet also. I don't know whether we will be able to address this.
Yeah, I think probably we should skip this.
Yes. The next is, do you accept this pandemic phasing out soon naturally?
I think that's really a tough answer, but I'm not trying to be again pessimistic. But we are facing a huge challenge. I think it's going to be around at least the next 3-4 years.
Yeah.
And the answer to this is to this lies within us. We need to be big tired human. We need to be going back to the basics about the cleanliness and all those kind of stuff that we needed to do that. So it's just like the more cleaner we are, the more healthier our environment is, the more healthier our body is, and the more fading away of this particular contagion from our bodies. So I think I have to admit that the next probably decade is going to be a huge challenge for us, and I don't think it's going to go out naturally.
Absolutely.
I think it's going to stay. I'm really sorry
Absolutely.
for this. I mean, this is my opinion, though.
No, I
Yeah.
understand. I understand. Next is, can't we artificially create the antibodies by studying and isolating from recovered patients?
Yeah, that's the other way around is, there's a lot of plasma intervention
Yes.
therapies going on, which
Yeah.
means from the recovered patients, we try to isolate serum plasma,
Yeah.
try to isolate the antibodies. So it's just like the platelets that we try to take from the healthy patients, which we call it as the STPs, single donor platelets.
Yeah.
In a similar way, we try to isolate the active antibodies present in the recovered patients and try to transfer it to the to the susceptible patients or to the patients who are really under treatment. So this research is going very healthily the last 2 weeks. People, researchers, scientists, and clinicians have really been successful in doing this.
Yeah.
And they have already transferred it into 3 patients.
Yes.
So 3 researchers, one from NHS, one from US, and one from India as well. People are trying to do this.
Yes.
The best example is from Boris Johnson, the UK Prime Minister. He was also he has also undergone this plasma intervention therapy.
Correct.
As believed. So yeah.
You're right.
Yes.
And I read yesterday from the government website that Kerala government the health department has applied to ICMR for testing with this plasma and the plasmid from the recovered persons. So they're trying to do that. Next question is also related to the same context. I think we already answered. The question is, shall we prepare convalescent serum from recovered persons or vaccine from recovered? I think already you have answered. Going to the next, there's only one more left. If SARS-CoV-2 has 99.1% similarity with SARS-CoV-1, then why still we claim we know very little about CoV-2?
That's the beauty of it. In analogy, I'll put it this way: why is that? The human genome is 99.99999% similar, and why is that? We're not stopping the diseases. So the question lies in the variant. So it all depends on the kind of systemic infection that particular virus is associated with, and that's where the system's genetics comes into picture. So so that 0.9% variation is certainly different from one isolate or one strain to the other strain. So there are about 132 assemblies, and among those 132 assemblies, the 99.1% to even the 100% similarity is absolutely associated with something called the nature of the variants that these are associated with. And these variants behave differently, and not just at the sequence level, but also at the structural level. And again, from the structural level, we need to ask whether or not these structural these variants at the structures are present inherently at the exposed site or at the buried site.
Okay.
So this is what we have tried to discuss in today's presentation.
Okay. And the final question is, what is your take on the statement, can virus be used to eliminate virus?
At this point of time, there are good bacteria that take out bad bacteria.
Uh-huh.
There are good fungi that take out good fungi.
Okay.
There are good viruses that do not take bad, but they try to help us understand whether or not this virus is really making a good story or not. What it does, how it functions.
Okay.
That basic understanding we know. But one pertinent question I would really try to put this is to answer this wonderful question. One take would be this. If you just imagine there are only less than 1% of viral biologists all over the world, and among the less than 1% of viral biologists, only 0.1 or 0.2%, less than 0.2% have the BSL-3
Facility.
Laboratories.
Yeah.
And not all BSL-3 laboratories, do a hell lot of research on viruses.
Yeah.
But they're absolutely more towards, understanding the pathogenesis or giving, medical reports to the patients,
Yeah.
from the hospital point of view.
Yeah.
So the onus is also on us, where we tend to bring out or help them at least identify the viable candidate, so that we can probably do some kind of research sitting at home or maybe in the lab, identify those particular viruses that could stealthily be discovered and be used as this particular target for the better interface of understanding the viral particles.
Okay.
So in conclusion, I would say that it's going to be a very risky affair to ask. I'm not saying that a wrong question or is a bad question, but in order to meet the answers for this particular question, we need to be prepared well at the diagnostic front.
I understand.
At the laboratory safety front.
Very clear. Very clear.
Very important. Yeah.
Very clear. So, Prash, if you don't mind, can I have a question? I know it was too much actually. So, this we were being discussing with my friends also. So, what the question here is, when COVID-19 came, everyone is behind it. Government, investors, hackathons, global hackathons, everyone is behind it. But hope you all remember that tuberculosis, TB, we have lost many more lives than this as of now. But still, there are many groups working on it, many investments have been done. But the seriousness and how the social media and the socio-economic things have seriously taken COVID-19, why TB is not taken in that context? What is your personal take?
Right. Even if it were the case, it's not just TB, Giri. I think that's a very good question. And even the basic, sanitation or basic, hygiene is not really considered, as a serious thing. I'm also, a bit surprised on this particular front. But in case of TB, there is a reason. This is again a personal opinion of mine.
Yeah.
I'll tell you from the scientific point of view and the non-scientific point of view. For TB, in fact, I've done an iota of research on TB as well, because I worked on hypothetical proteins.
Okay.
The proteins now which tend to be characterized, but their function is not ascertained properly.
Yeah.
Mycobacterium harbors about 42% of hypothetical proteins. From the scientific perspective, all researchers never targeted these hypothetical proteins. They only targeted those proteins that are that are the rest, the 8% proteins that are well known. And they somehow they never believed in drug discovery phase. Even they tried to adopt certain kind of drugs or identification of drugs. They thought that that's not their cup of tea, and the drugs were absolutely very, very expensive.
Yeah.
From the non-scientific point of view, since from Indian perspective, from Indian orthodox perspective or unorthodox perspective, right from day one, our ancestors, kept all these diseases at bay. They said that yes, you might get, Mycobacterium tuberculosis infection, so please stay away from me.
Yeah.
Okay, so you might do this. So a lot of people, kept these so-called people who are prone to these particular diseases, away from their houses or away from their villages as well.
Okay.
They're not really considered, those people, they're not even considered as human. That's a very unfortunate thing.
Yeah.
So in that way, the mindset has prevailed in such a way that yes, TB is such a scary disease. Let's forget that. Let's not do any research in this. Even the scientific mindset also has really gone, dividends.
Yeah.
And the third aspect to this is more towards the economic perspective. Unfortunately, all the researchers, with due respect to them, they only thought what is in for me at the monetary level.
Everywhere.
I
Yeah.
come up with a diagnostic kit. I come up with
Yeah.
an interface of mycobacterium infection, and maybe come up with a TB diagnostic kit. How much I could earn. So if an entrepreneur starts thinking in that way, then the nation is in jeopardy. So that is an unfortunate thing, and that has really happened with with India. But amazingly, I've got a very good friend of mine named Jagdeep Singh Sohal, who has done extensive research in mycobacterium.
Okay.
He has developed a wonderful kit along with Professor Ratnagiri for mycobacterium avium. And they're all for cattle, because many cattle are infected with mycobacterium infection as well. But that knowledge was never translated into human. I don't know why. But I think these three reasons are very important for us. I think from the scientific and from the unscientific perspective, and the third one is from the industrial or from the more economic perspective. These are the kind of challenges. Maybe if we can overcome these challenges and ask a question that yes, we are absolutely good. We are entrepreneurs. We need to develop kits, but at the end of the day, we need to do some kind of human service to the nation.
Yeah.
It's not just developing kits and making money for us, but also what is it intake for the society.
Correct.
If we can ask these kind of questions, then I think we are going to win the battle. Yeah. So that is my point.
Thank you so much for that wonderful answer. If you don't mind, I have one question came up, but sorry, there are two questions coming up, but I don't think we can answer everything. We have already given you time, so you decide. Virophage technology can be an answer for this COVID-19 situation.
Yeah, I mean, that word I also heard, pretty recently. The bacteriophages are nothing but the viruses, invading bacteria. The virophages are, the viruses, the phages, infecting the viruses again. That has been, there. But for that, I think, that is going to be very, very, risky thing. And I think, not all labs would really be, interested in this. I don't know, but I think that's going to be a bit risky if you ask me, from a biologist's point of view. Yeah.
So earlier you said that COVID-19 will not phase out naturally. It might take some time, on your personal opinion. But do you think whether the SARS got phased out or it is still there?
It's still there. I mean, that's where even if you see, there were few cases reported where people thought that it was novel coronavirus, but it was not novel coronavirus. It was SARS-CoV. So there were reports, even in Wuhan as well, even in some reports in Italy. Maybe in that particular thing, there are certain probably periods present in such a way that the contagion would behave ephemerally in both ways. It has both HCoV infection and as well as novel coronavirus infection as well. And I don't think SARS-CoV has faded away. In fact, it has spread its wings. It has had a lot of dividends, and it's still occurring in many nations as well.
Great. Thank you so much, Prash. So I'm just going for conclusion. Let me tell you a few things and then we'll conclude it. So hi every attendees, and thank you so much for your patience. I know we crossed even the lunch time also. Too much energy for Prash to stay back with us. Of course, you need a lot of energy during the lunch time to stay back. Our next sessions are happening on Saturday at 5 p.m. on polypharmacology. And also on Sunday, April 19th at 5 p.m. on Autodoc, a training demo session. So it's just wanted to inform you. And I would like to take this time to thank Prash. Of course, we had some technical glitch, which is expected, and nothing to worry about it. And thank you all for joining us and patiently listening us with questions. Around 23 or 24 questions were there altogether. And thank you so much, Prash, for taking so much energy to replying to each and every questions patiently. And I am sure that everyone is convinced with your session, and looking forward, and wishing you all the best for your studies, which is pilot studies, to continue. And we are looking forward to something, a very good outcome from there.
Oh, thank you so much, Giri, for your wonderful, thoughts, time, and energy, and thank you for inviting me. I really like the way, the participants ask the questions.
Yeah.
I think this should be the case, and I think we should not stop asking questions.
Yes.
I really like this kind of environment, especially seeking questions, answering the questions and
Yeah.
learning. Thank you so
Because
much.
our intention is more of interactive sessions on answering questions rather than presentation. So we want to make the presentation like for 3 to 4 slides or maximum 6 slides, so that it will be more productive. Anyway, thank you so much, everyone. Have a good rest of the day, and